Good afternoon, everyone. My name is Amit. Thank you so much for having me and organizing this event. So special thanks to Hideki, and thank you to AWS for organizing this event. Um, Japan is actually one of the earliest leader in genomics. When the Human Genome Project started in 1990, Japan was one of the key countries, together with US and Europe, in starting the genomic revolution. So they are one of the earliest pioneer. Especially in Japan, the Riken Institute was one of the first institute to be a big part of the Human Genome Project, and they helped sequence the first human genome. And for Japan, the special focus was chromosome 21. And as you know, chromosome 21 is involved in the Down syndrome. So the Japanese scientists found the first sequence of chromosome 21, which is now used in every place. And even today, Japan hosts one of the biggest databases for genomics data, which is the DDBJ. Any public data set that any scientist creates, it is uploaded to DDBJ for other scientists to upload. So Japan continues to be a big innovator and a big support in the genomics ecosystem. So we are very excited about an opportunity to work with more organizations in Japan. A little about me. I am a computer scientist by training. I have a PhD in machine learning, but the application of machine learning and programming that I found most challenging and meaningful is in the medical domain, because the data is very messy, but the impact is so high. So after completing my PhD in computer science, I worked in a cancer research lab at Harvard. And the reason they had hired me is that genomics had just become commercially available. NGS had just become commercially available. So very few organizations had the knowledge and the resources to do these analysis. So my job was to figure out how to analyze genomics data for cancer research, how to present the data, how to store that, and so on. So I was, in the white shirt, the only computational person in a lab of 20 bench scientists, people who do the actual experiments. And I would support my colleagues in doing their data analysis. Because as you know, many bench scientists, they are not trained in programming. They are trained in doing lab work, experiments, but they often don't know programming. And with NGS genomics, the data size is very large, and analysis can be very complex. So it becomes very challenging for them to do the analysis on their own. For simple things, they can even do the analysis in Excel, create a simple box plot and stuff. But for genomics, the data size is huge, analysis is very complex, so they need to depend on a bioinformatics person like me to do the analysis for them. So I had a very productive time working in the cancer lab. And this is where I learned more about biology, about genomics, and how it can be so helpful. So of course, one big application is cancer itself. So in our lab, we used to study how cancer is started in a human being. So generally, many cells, they become terminal cell, but because of epigenetic changes, they become cancer stem cell. And then that cancer stem cell gives rise to a complete cancer that we see in the clinic. So all that can be studied very easily using genomics. And using genomics, we can see which genes and which biological pathways are responsible for creating the cancer in us. And once we identify that, then we can discover drugs which can actually target those genes or those pathways. So this is why, as a computer scientist, I saw how powerful genomics is in discovering cancer, finding new cures. And now there is a new technology called cell-free DNA. So even before the symptoms of cancer is visible to the doctor, there are trace amounts of DNA floating in the person's blood. So using cell-free DNA, many companies are now offering early detection of cancer. All this is possible through the power of genomics. Another field where we are seeing a lot of impact of genomics is reproductive health. For example, we can start with carrier screening. If two people plan to have a baby, the baby may have some disease if both the people carry recessive genes. So now one can do carrier screening, both the father and the mother can be screened, and you can predict what disease a child is likely to have. So you can make some decision early on, you can be more prepared early on. Another application is in IVF. So in IVF, in one cycle, we have 6 to 8 embryos, and you take one embryo and you implant it in the lady. The success rate is only 30%. But if you use genomics and find the best embryo, then the success rate goes from 30% to 60%. So a huge benefit in doubling the chances of pregnancy in IVF using genomics-based screening. Another example we have seen with during the COVID virus. If you do a PCR test, you only have a binary answer. Is there COVID? Is there no COVID? But with sequencing, you know the exact variant of COVID. Is it the Delta variant, Omicron variant? In fact, genomics is even more powerful. What many countries were able to do, they took the sewage samples, all the garbage samples, and they were able to detect coronavirus in those garbage samples. And they are already collected by the municipal, so the sample was available. So using that, they are more accurately able to predict when the corona has come in a city, how is it growing, is it going up, is it down. All this is possible only with genomics, not using any of the other traditional techniques. So genomics is a truly powerful new technology, and it is transforming medicine. But there is a challenge. In this graph, we see in the line in the red, the price for genomics is growing down exponentially. It used to cost hundreds of billions of dollars to do sequencing of one person. And now that cost has come down to less than $200 to do whole genome sequencing of one person. So then the graph in the red, you see that the price of genomics is going down exponentially. But because the price is growing exponentially, people are doing more and more sequencing. So if you look at the line in the red, in green, the amount of data is also growing exponentially. It is estimated that within 5 years, genomics will have more data than YouTube or Twitter or all of those channels. Today, hardly a few million people are sequenced. Going forward, you can imagine the price of sequencing will go down to less than $100 for full genome. Then billions of people in the world will be sequenced. And that will generate hundreds of exabytes of data. So even today, Amazon will struggle to store. If every human being on the planet gets sequenced, maybe Amazon cannot even fit all the data in S3. So Amazon will also grow its resources with time. And then the biggest challenge is we have this massive data set, billions of people will get sequenced, but there is a disconnect. The people who do the experiment, the doctors, the scientists, they don't know programming. They don't know how to handle terabytes of raw data. So then they depend on bioinformatics people like us who do the analysis for them. So this process becomes very slow, very inefficient. So this is what I experienced working in the lab. All my colleagues, they did the experiments, they generated the data, and then they had to come to someone like me to do the analysis. And this problem is really holding back genomics. If you do a survey and you ask people, what is the biggest bottleneck for adoption of NGS? Why aren't more people using genomics if this is so powerful? So there are so many steps involved, collecting the sample, doing the library prep, the actual sequencing and data analysis. In every survey we have done, the number one reason stopping people from more genomics is always data analysis. It is the biggest bottleneck. The cost has come down, the technology has become easy to buy, but the data analysis and visualization and interpretation continues to be the #1 challenge in genomics. The way most organizations try to do this is it's easy to buy the sequencing machine. It costs a few $100,000. You can buy the sequencing machine. It's like a large printer. You set up the lab. And the sequencing machine generally, if you load the sample, it will give you the raw data in 1 to 2 days. So that part has become fairly simple. But once you have the raw data, then what organizations do is they buy a lot of these on-prem computers, and then they hire all these bioinformatic analysts to do the analysis. This is where the delay happens, because now the scientists, they cannot do the analysis. So they have to depend on this in-house team to do the analysis for them. And these team are usually doing the analysis manually, so it takes a lot of time for them to come to each project. And because you are doing on-prem, and these are very big computing jobs, so then there's a backlog. You have to wait in the queue. You can only run 5 jobs at a time. You can only store so many terabytes of data. So finally, the analysis is done, and the user gets some static files like PDF or Excel. So not just this process is very slow and expensive, but the biggest problem in this model is that for the scientist, the whole thing is a black box. They have the raw data, they get the final result, but they don't understand the process. How did we go from the raw data to the final result? And in my experience, this is a huge drawback because scientists understand the most about the data. If a physician is treating a patient, they understand the most about the patient. Bioinformatics people can do the analysis, but they don't have the scientific knowledge. They don't have the clinical knowledge that the biologist or the physician see. So in our view, the analysis should be done by the scientists. We need to make the analysis so easy that scientists, physician, they are able to do the analysis on their own. Because then they are able to gain so much knowledge from the data if they see the whole process from the start to the end, not just a final result. Another big reason why this process becomes very challenging is science is not iterative. You ask one question, you get the answer. You look at the answer, it gives rise to more question, and then you have more answers. So there's a lot of back and forth involved in any scientific process. In the current model, every back and forth takes weeks of delay. So you have a question, you go to the team, they do the analysis for you, you get the result back, you think over it, you have more question, and then you go back to them. They again take a few more weeks, and then they come back to you. So this process is very, very inefficient and very frustrating for both parties. Because even bioinformatics people, they want to do something in machine learning. They want to do cool, complex analyses. It is frustrating for them to keep doing simple, small analyses over and over again. So they get challenged by this model as well. So what we believe the way to go is really to empower the bench scientist. Even though genomic analysis is very complex, I saw this opportunity, so that is why I left academia and I started this company, because I felt that there is a need for software which can really simplify a very complex problem. Make it so simple that any bench scientist, any physician, with only a few minutes of training can do a complex analysis herself. And not just do the analysis, but they have a nice dashboard where they see all the data, visualization. If they have a question, if they want to change the analysis parameter, they make the change, and then the data updates very quickly. So now we are truly democratizing genomics, giving the power in the hand of the bench scientist. So that is our vision for the future of genomics. So I'll pause here. Thank you. If you have any questions, any comments, happy to discuss.